everyone, and welcome to another exciting seminar in our series. Before I introduce today's speaker, I just want to quickly remind everyone that, as always, there will be a Q&A session after the talk. If you have any questions for the speaker, you can post them in our designated Slack channel, where you can also upvote the questions that you would like to see answered. Our speaker today is Professor Walter Salzburger from University of Basel. Walter received his doctoral degree in zoology from University of Innsbruck. After a postdoctoral fellow position at University of Constance, he joined the University of Lausanne as a junior group leader. He soon after moved to University of Basel, where he was first assistant professor and now full professor in the Zoological Institute. Walter and his group's research is focused on understanding the processes that underlie adaptation, speciation, and evolutionary innovation, studying in particular the exceptionally diverse adaptive radiations of African cichlid fishes, which is also the topic of his talk today. So with that, thank you very much, Walter, for accepting our invitation, and over to you.
Thank you very much, Julia, for this kind introduction. Thanks to everyone from the team for setting up and for organizing this amazing online evolution and ecology series of seminars. And today it's my pleasure, as Julia said, to talk about some aspects of our latest research on the dynamics of adaptive radiation in African cichlid fishes. Some of those you have seen in the background morphing into each other. And in the more broadly sense, more broadly speaking, our research wants to explore in a more general way how sort of the way how organisms diversify. And in that sense, how biodiversity comes about. Well, clearly, diversification is a ubiquitous evolutionary phenomenon. Even in that tiny little branch in the tree of life that leads to our own species, we do find a great deal of diversity. Diversity in body morphology, in body size, in coloration and pigmentation, in locomotion, in behavior, etc., etc. And since Darwin and Wallace, for that matter, we do know that selection plays a crucial role in the origin of species. What is perhaps less well understood is how natural variation as a substrate for selection comes about, and which environmental factors, but also biological features are more conducive to diversification than others are. Now, I guess all of us would have a lot to say and perhaps a lot to like about Charles Darwin. Personally, I do like a lot that Charles Darwin started out his career as a naturalist with some field work. Well, more precisely, I should say that he spent 5 years on board of the HMS Beagle on a journey that literally brought him around the globe. And there is sort of this spot on that journey that he visited that remains somehow in our collective minds, at least when it comes to Charles Darwin, and that's of course the Galapagos archipelago, and with it the finches that Charles Darwin collected on this island. Darwin's finches, as they are called, are today perhaps the most famous example of an evolutionary process called adaptive radiation. That is the origin of a great deal of ecological and morphological diversity from a common ancestor, and in a short period of time as a consequence to the adaptation of distinct ecological niches. Now, in a more general way, it is believed that adaptive radiations are responsible for a great deal of the ecological and morphological diversity of life on Earth, as illustrated perhaps by another example of adaptive radiation, the so-called Cambrian evolutionary radiation or Cambrian explosion, during which, in geologically speaking, no time the main body plans of the animal phyla have evolved. At the same time, this Cambrian explosion illustrates that species emerging from an adaptive radiation, like sort of this weird creature here called Anomalocaris, are not or may not be very long-lived, because what happened relatively soon after the onset of the Cambrian evolutionary radiation is that there was a Cambrian extinction event in the late Cambrian, and what we see here is sort of a reconstruction of the extinction rates over geological times by means of estimations from the marine record. And here we see this late Cambrian extinction event that led to the extinction of a number of creatures that evolved in this Cambrian explosion. And just note that this late Cambrian extinction is not even one of the big 5 mass extinction events that have occurred since. Now, on a macroevolutionary scale, we can say that the diversity of life on Earth is shaped by the interplay between adaptive radiation on the one hand, creating organismal diversity, and extinction events on the other hand that take diversity away. Now, what makes something an adaptive radiation? Well, one characteristic feature of an adaptive radiation is clearly common ancestry. That means, as I said before, an adaptive radiation starts out from a common ancestor, as shown here by some cichlid species from Lake Tanganyika. And this very often gives an adaptive radiation somewhat of a direction from a more generalized species, a generalist that starts an adaptive radiation or initiates it, towards specialized species. Now, another aspect of adaptive radiation is clearly that radiations happen quickly, that speciation, the origin of species during adaptive radiations, is rapid, which is not always easy to measure. So very often this is determined by comparison to outgroup species, as we do see here again with the cichlids, where we have species per outgroup lineages and then an adaptive radiation event that produces a great deal of diversity in no time, as I said before. And this is why we often, at least in the case of the cichlids, call this even explosive speciation. Now, one key characteristic feature of adaptive radiations is the correlation between phenotype and environment, meaning that the phenotype of species within an adaptive radiation match the specific ecological niche to which they have adapted to. And I would like to show that here with the Caribbean anoles lizards, where the different species show a very characteristic body plan, including certain limb lengths and sort of a massiveness or non-massiveness of the body, depending on where they live on these islands, be they on the ground, on the trunk, on the twigs, and or wherever. And finally, what is characteristic for adaptive radiations is that the different species differ in phenotypes that are useful in the context of exploiting particular ecological niches. And here I would like to go back to Darwin's finches, where it's kind of obvious that the finch with the massive beak is the one that is better adapted to crack open hard shell seeds. And with the cichlid fishes, especially those of Africa, the anoles lizards of the Caribbean, and Darwin's finches, I have also introduced to you sort of the main examples of adaptive radiations in vertebrates. Now, as we heard before, and as I said before in my group study, cichlid fishes, in particular the African cichlid fishes, and to many people, these are really the hallmark of adaptive radiation. I would like to quote just a few people here that have not worked with cichlids, so that are kind of biased in their view about cichlids. Dolf Schluter, for example, puts the cichlids together with some other adaptive radiations and the dinosaur extinction and the origin of our own species among the most celebrated events in the history of life. Coyne and Orr say that no radiation is more impressive than that of the cichlids in Africa, and Mike Arnold in his book says that it would actually be an understatement to say that cichlids are astounding in the level of diversification. Now, why would people like that put the cichlids sort of on top of perhaps many other animal model systems when it comes to study diversification? Well, the reason is that they are diverse, and diverse with respect to many features and many aspects of biological organization. First of all, there's a lot of different cichlid species. If we just sort of zoom in the East African Great Lakes, where many cichlid species and perhaps the most famous radiations of cichlid fishes occur, then we will find that in the area of Lake Victoria, about 700 different species have evolved, and in this case, in not more than 100 to 200,000 years. Lake Malawi contains an estimated number of 800 to 1,000 different cichlid species, and in Lake Tanganyika, there are roughly 250 cichlid species. I will get back to these species and numbers, particularly for Lake Tanganyika, a bit later. But from an evolutionary perspective, perhaps even more amazing is that all these species are endemics. That means the 800 to 1,000 cichlid species of Lake Malawi only occur in Lake Malawi. They have evolved there. They do not occur anywhere else on the world. Now, it's of course not only the sheer numbers of species that make these cichlids so unique. You already see on this compilation that they are highly diverse when it comes to their morphology, especially when one considers the ecomorphological traits of these cichlids. In basically any of these cichlid adaptive radiations, we do find all kinds of different mouth and head morphologies specifically connected to a particular feeding strategy. For example, there's this insectivorous species, often pointy teeth, so that they can pick out insects from the ground. There's zooplanktivorous species with pipette-like mouths so that they can suck in zooplankton. There's all kinds of piscivorous species, some of which are torpedo-shaped so that they can hunt other cichlids. There is algae scrapers, there is leaf eaters that feed leaves that fall into these lakes, there's mud diggers that shovel up mud and filter feed it through their gill raker apparatus, there's sucking species, there are species that bite off fins from other cichlid species, there are eye-sucking species, and there are the bizarre scale eaters that exclusively feed on scales of other cichlids, and these are the ones, at least some species in Lake Tanganyika, that have this polymorphism in mouth morphology, whereby about half of the individuals from a population have a mouth that opens to the left side, the other half to the right side, so that they can better and more efficiently attack their prey from behind. But sort of the level of overall morphology of the head and the mouth apparatus, the so-called trophic apparatus, is not everything that cichlids have to offer in the context of feeding adaptations, because cichlid fishes have evolved a way more complex feeding apparatus, and in order to understand that, we have to look into a cichlid's head here on the basis of a computer tomography scan, because cichlids, in addition to the oral jaws that we see here in pinkish coloration, have evolved the so-called pharyngeal jaw apparatus, and this is a bony structure derived from the fifth branchial arch, and this is an efficient feeding apparatus consisting of a lower pharyngeal jawbone here in more greenish coloration that looks like this, that sits in the pharynx, and its counterpart. And with muscles attached to that, this is a highly efficient machinery to process food. So in a sense, it is believed that cichlid fishes have decoupled or that a decoupling is going on between the oral jaws that are there for food uptake and the pharyngeal jaws that are there for food procession. And we see that here now with a few real images where we always see a fish head from a real cichlid fish from Lake Tanganyika, and the associated pharyngeal jawbone, the lower pharyngeal jawbone in this example. And what we do see here, for example, is there is a species with very massive pharyngeal jaws and big teeth. This is a snail crusher, whereas next to it is an algae-eating species that has a lot of fine teeth, which obviously work well to macerate plant material. So we have a lot of species, we have ecomorphological diversity, but we also have a great diversity in color and pigmentation patterns, and this is why many cichlid species are fancied by aquarists that like to keep them at home. Very often we find the situation that in cichlid species we have very colorful males, like this one here, and relatively dull females, and that hints, of course, that some sexual selection mechanisms are going on in these cichlids based on female preference mechanisms. Now, cichlids also show a great diversity in behaviors. I would like to illustrate that here with the breeding behavior of cichlids. Many cichlid species in East Africa are mouth brooders, where the females or the males or in both parents keep the babies in their mouth until they're sort of free living. Other species use structures like as empty shells. We see one species from Lake Tanganyika here hiding from the photographer, but they don't only use them to hide, but they especially use them to breed and raise their families. So sort of the empty shells as their houses. Again, other species are substrate spawners, like the biggest or one of the biggest cichlid species in the world. Here, Bulocheirochromis from Lake Tanganyika, that has several thousands of fry around it, and the two parents guard these offspring for a long time until they're almost mature, and guarding in this case often also means defending it. So here we see some sort of aggressive behavior towards the photographer. Finally, also the genomes of cichlids have been explored some time ago in the framework of the first and initial cichlid genome paper. And one question in this project has also been sort of what might be specific for the cichlids on the genomic level. And it has been suggested, first of all, that diversification in cichlid fishes primarily involves non-coding divergence, which makes sense because of the rapidity of diversification. It has been suggested that especially the radiating cichlid lineages of the East African Great Lakes compared to the tilapia and some outgroup species contain novel classes of microRNAs, basically to facilitate gene regulatory processes. It has been shown that the radiating clades show, or the genomes of those contain more gene duplications. The rate of positive selection is greater, and also there is different kinds of and higher rates of repeat element insertions in their genomes. And in general, the genomes have been shown to be more diverse than expected by the sheer age of the lakes, which has been interpreted as a signature of standing genetic variation. Now, against this background, we have some years ago set out to explore the cichlid fishes of Lake Tanganyika. Here you see an image of the shores of Lake Tanganyika. Here you see another image with a boat that we have used temporarily as a research vessel to visit many different places on the lake and dive underwater to find cichlids and see them. And we have specifically chosen Lake Tanganyika because, in my opinion, and in the opinion of a few other much more famous researchers, especially Fryer and Als, have said that taken as a whole, the Tanganyika cichlids represent the most outstanding example of adaptive radiation of all to be considered. Well, as I said, we have set out to collect Lake Tanganyika cichlids, and we have managed over several years to collect basically each and every species of cichlid fishes that exists in this lake, very often while diving and using this gill nets, and here you see my colleague Fabrizio Ronco and myself in our collection, because we have established a collection of cichlid fishes, a new reference collection here at our institute that contains basically all different species of Lake Tanganyika. These are jars like that, so that you have sort of a size measurement to this image. So they are relatively small, you may say. Now, based on this collection and our accounts of cichlids in the field, we could first of all make an inventory of the cichlid fishes in Lake Tanganyika and sort of taxonomic accounting, including both described and undescribed species. So most of the diversity is described, and we have come up with a relatively good estimate for the number of species in Lake Tanganyika, at a bit more than 240 species, whereby members of 3 groups, these are the ones here in gray, this is 1 species each, are cichlids that belong to more ancestral lineages that have colonized Lake Tanganyika at a later stage. But more importantly, our collection has been used to sort of phenotype, ecotype the different cichlid species in the lake, and to do much more on that. And you see here again Fabrizio Ronco at our computer tomography scanning device. And what she has been doing, for example, is that she has scanned 5 specimens per every species with this CT scanner, so that we can obtain precise measurements in 3D of this pharyngeal jaw apparatus that I've been introducing to you before. We have also phenotyped body shape based on 2-dimensional X-ray data. We have collected information about pigmentation patterns, in this case in male individuals. For sort of placing the species in an ecological context, we have used stable isotopes, in particular the carbon and nitrogen stable isotope, that we can determine based on small tissue samples from muscle tissue. And this allows you to, relative to one another, place the different species in a food web. So the kind of logic is that the nitrogen accumulates across the food pyramid, from algae-eating species or plant-eating species up to predators, whereas the carbon axis informs more about near-shore versus offshore habitats. And we have sequenced a male and female genome for every single cichlid species in that lake, plus some outgroup species, leaving us with a very nice integrative data set that includes genomic information, ecological information, and morphological information. With the genomes, for example, we could produce a genome-wide phylogeny of the cichlid fauna of Lake Tanganyika, which you see here, whereby the different colors indicate the sublineages that have evolved, the sublineages of cichlid fishes that have evolved within Lake Tanganyika. They are called tribes. But coming back to what I said about the features of adaptive radiation, so we might interpret common ancestry in a sense as monophily, and indeed the adaptive radiation of cichlid fishes in Lake Tanganyika, except for those ancestral lineages that I have mentioned before, indeed go back to a common ancestor. But of course, we were also very much interested in the timing of this radiation, and especially whether it's compatible with in situ adaptive radiation, that means inside Lake Tanganyika, so within the geographic and temporal confounds of Lake Tanganyika. And in order to figure that out, we actually first had to zoom out a bit of Lake Tanganyika and have a look at cichlids on a more global scale, because cichlid fishes are in fact distributed on land masses that used to belong to the supercontinent Gondwanaland. So the current distribution of cichlid fishes is shown here, with representatives in North, Central, and South America. Then some representatives in Madagascar, India, Sri Lanka, and of course the African ones. And what we did, we included representatives of all these sort of areas in the world in our genome sequencing project, because we wanted to place the global diversity of cichlids in a larger phylogenetic and phylogeographic context. First of all, to get an idea about the timing of cichlid evolution globally and especially in East Africa, but second of all, also to learn about the history of cichlid fishes as a family and to distinguish between three sort of main hypotheses about cichlid evolution in the first place, namely Gondwanian vicariance. That would mean that the ancestor of all cichlids already existed on the supercontinent Gondwanaland about 150 years ago, or whether the current distribution is the product of either oceanic dispersal, so transoceanic dispersal, or even perhaps independent colonization. And Michael Machina, who led this study, invested an incredible amount of time and in even coding new software to place the new cichlid genomes representing the diversity of cichlids globally into, as I said, a much larger phylogeny of fishes where there were abundant fossils. And with this, he could produce a time tree of cichlid evolution, a genomic timeline. And you see here that we can estimate that based on that, these kinds of analysis, that the diversity between South American and African cichlids occurred about 65 million years ago. So clearly the Gondwanian hypothesis can be refuted. But for us and for Lake Tanganyika, the most important thing is that we could use this genomic timeline of cichlid evolution now to put a date on our cichlid phylogeny, the genome-wide phylogeny that I've been showing before. And we get an estimate of about 9.7 million years for the origin of the Lake Tanganyika cichlid radiation. And this is pretty much the time when Lake Tanganyika started to form. So we have an independent estimate that is not dependent on the geological age of Lake Tanganyika itself, but on fossils from many other groups that are non-cichlids. And we still sort of get a scenario that is compatible with in situ radiation, and that basically means that the 250 cichlid species in Lake Tanganyika, which are organized in 52 tribes and genera, really evolved within Lake Tanganyika in the last 10 million years. Now, a second major feature of adaptive radiation is, of course, phenotype-environment correlation. I used the anoles lizards here to explain that to you. And so we also want to test it for that, of course, in the cichlid adaptive radiation in Lake Tanganyika. And here really the data on morphology and ecology came in handy. I'm only showing you as an example the so-called morphospace that is based on these three-dimensional measurements of the pharyngeal jaw apparatus of the different cichlid species. And what we see here is a huge data cloud, obviously, whereby each polygon that we see here is the 5 specimens that were scanned from a particular species. And this data cloud is separated according to the 2 main principal components that we can actually interpret very well according to morphology. So PC2, for example, here is loaded from pharyngeal jaws with long horns to very sort of thick and those with very short horns. PC1 is mostly also going from very broad pharyngeal jaws to sort of very thin ones. And what we see here is the data from the different species color-coded again according to these sublineages, these tribes. And maybe one aspect that is interesting here is that what we see here, that members of different tribes can often be quite similar. In this case, we have in gray one group here, in pink one group here. So they have very similar jaws, but they also are phenotypically similar, at least in terms of body coloration, because these two, even though they belong to different sublineages within the radiation, are both relatively large predatorial cichlid species. So they also show similar feeding behaviors. And maybe another observation from that graph is that apparently, at least when looking at that, those species, or those tribes that contain more species, take a larger fraction of the morphospace, and we have tested for this observation. This is really the case. So you see here the number of species in log scale from up to 100 species in the pink group. Here, sort of the morphospace extent. So in a sense, it's really the case that species-rich groups also occupy a greater area of the morphospace. This shouldn't really be a surprise, but it tells us that sort of the unit species in that case makes some sense. As an environment correlate, we have used, as I said, the stable isotope data. Here you see data based on 10 specimens per species with the 95% confidence intervals shown. And also these data we can interpret very well from on the carbon scale, nearshore to offshore habitats. And as I said, on the nitrogen axis from plant eaters or algae eaters over invertebrate eaters to predators. So we can distribute we can sort of disentangle the different species very nicely on the carbon and nitrogen axis. Maybe an interesting side story, this one who is kind of a top predator on the nitrogen scale in orange. This is in fact a scale eating cichlid, so that apparently also eats scales of fish eaters. It's the highest on the nitrogen scale. But of course, we were interested in the correlation between phenotype environment data. Well, sorry, I forgot to say that also here, species-rich tribes occupy larger fractions. So we were really interested in sort of the correlation between phenotype environment data, and this is what we see here for body shape, because if we sort of look at the body shape axis and compare it to the stable isotope projection axis, we do see a very strong correlation between phenotype and environment. The same we do see with mouth morphology, so the oral jaws that we can again interpret well from very pointy mouth here to very beak-like mouth of the algae scrapers, and also in the pharyngeal jaw morphospace we find a very strong correlation between sort of the phenotype, pharyngeal jaw phenotype, and the stable isotope data. Right. So the next step was that we were actually interested in the dynamics of adaptive radiation. So how the different species, but in particular the ecological and morphological diversity evolved. And in this case, now we combined the phylogenetic information from the genome-wide phylogeny that I have shown you before with the trait data. But instead of just mapping the different trait data on the phylogeny, what Fabrizia Ronco did, who led this study on the dynamics of adaptive radiation, is to sort of time slice through the phylogeny and reconstruct for each trait the morphospace along these timelines from the present to the past, so from now towards the onset of adaptive radiation about 10 million years ago. And for each trait then, or each trait complex, we should better say, we can then sort of compare the actual morphospace evolution to an expected morphospace, for example, expected under neutrality. The proper model would be Brownian motion. That would mean that, that the general thing that actually disparity, so difference between taxa, should accumulate more or less linearly over time. And why would we compare that or actual trait evolution in an adaptive radiation to something like a Brownian motion model? Well, this is because there is existing theory about adaptive radiations and how diversity should evolve within an adaptive radiation. And one very often presumed scenario about diversification during adaptive radiation is what is called the early burst model, whereby disparity, typically morphological disparity, is said to accumulate more towards the onset of adaptive radiation until the major niches become filled so that it slows down then. Other people believe that this is also possible at a later stage, which would perhaps look like that. Whereas, of course, if a trait evolves gradually or more or less gradually during time, then we would find such a curve. And this can now be relatively easily be shown, especially in the comparison between real data and models, by measuring the delta between the real data and the Brownian motion simulations. And for example, for an early burst or for a late burst, we would see that for an early burst, such a signal, and this would be a signal for more or less gradual morphological evolution in the course of an adaptive radiation. So we did all that for 4 different trait complexes, body shape, oral jaw, and pharyngeal jaws we have seen before, but we also introduced here our data matrix on pigmentation traits for the simple reason that pigmentation has also been speculated to play an important role during at least some adaptive radiation. So diversification in pigmentation pattern should also contribute to diversification. And the first thing now that I'm going to show you for these 4 different trait complexes is how the morphospace or the ecomorphospace became filled throughout the course of the adaptive radiation, starting here about 10 million years ago at the onset of the radiation until today. And what is shown here is basically using the combined information about relatedness and the morphospace dimension. We basically reconstructed in these time slices along the phylogeny the extent of morphospace occupied for each of these trait complexes. And what becomes clear is that there is differences. I'm going to show you here now the time point at which 50% of the morphospace were filled. So clearly the filling of morphospace was going on earlier, at least the 50% filling in body shape as compared to, for example, pharyngeal jaw shape. Now, in the next column, I'm going to show you exactly these simulations against the null model of Brownian trait evolution that I've explained in the previous slide. And what we see here is that in each trait, we could find discrete, what we call pulses of ecomorphological diversification throughout the radiation, so that there was not something like a gradual evolution in these different trait complexes, but obviously there were phases of accelerated evolution, as we could also see when comparing evolutionary rates. And the interesting thing is that we can pinpoint 3 different such pulses. A first phase in which a pulse in body shape and a little bit in pharyngeal jaw shape occurred. Then a second pulse primarily driven by diversification in oral jaw morphology, and then finally a third phase with a pulse each in pharyngeal jaw diversification and in pigmentation patterns. And this last one is one that is connected to the increase in number of species that we do find in this cichlid adaptive radiation. Now, given all this information that we had collected on the cichlid fauna of Lake Tanganyika, we could also look into one aspect of diversification that has to do with species richness, because let's go back to the phylogeny, now not anymore in the circular, but in a rectangular way. One observation that I've kind of mentioned already is that in these subgroups of the Tanganyikan radiation, the so-called tribes, there is great differences in the number of species they contain, even though they all go back, so all of these species go back to the same ancestral species about 10 million years ago, and that they all evolved in exactly the same lake, exactly the same environmental condition and possibilities and opportunities whatsoever. There is some groups that have a lot of species, in particular the so-called lamprologines that have evolved 100 different species in that time, whereas others, as I said, with the same starting point and conditions have evolved way fewer species. So an order or 2 orders of magnitude difference between these sublineages. And so we thought we can use this setup to ask the question now, what is really different between the species-rich lineages, on the one hand, and the species-poor lineages on the other hand. And of course, we digged into the genomes to see whether we can find any particular feature that is especially in the genomes, well, with a focus on the species-rich lineages. And of course, we had some hypotheses for that, namely from this initial Cichlid genome paper. So we looked, for example, into the transposable element contents and the diversity of those. And apparently, if we look at the data, here the species poor, here the species rich ones, there is no difference or no correlation between transposable elements and species richness. The same is true for the number of gene duplications that have occurred in the different lineages, and also with respect to signatures of positive selection, we could not see any differences. However, we figured out that the species-rich lineages contain species that are on average more genetically diverse as measured by the per genome heterozygosity. So this is in a sense for the first time a clear link between genetic diversity and species richness in the context of an adaptive radiation. One thing that we can say is that this is not a direct consequence of hybridization. Many people believe that adaptive radiations might be triggered by hybridization. So, and heterozygosity in a sense can easily come about, or increased heterozygosity can come about through hybridization, but at least there is no direct evidence, because if one looks at hybridization signatures, then there is no such strong correlation, but it turns out, and we kind of hinted to that by a simulation, that the combined effect of phylogenetic history of a clade of such a tribe and the rates of gene flow within the tribe versus non-existing gene flow between tribes can produce a pattern that is in heterozygosity that is similar to that. Now, I would like to make one final point back to or in the context of this question of diversification, because in a parallel study to those that I have presented so far, that study was led by Atimed El Taher, and she looked at expression profiles and the gene expression dynamics of the different cichlid species in that radiation. So we picked about 1/3 of the species of the radiation that are phylogenetically representatives, and took these 6 tissues, and for each species we took 3 males and 3 females, ending up in a total of more than 2,200 expression profiles for the radiation. And so we sort of looked at the gene expression changes that occurred in or during the course of the adaptive radiation of cichlid fishes in Lake Tanganyika. I would just like to show one thing, namely, if you make a pairwise comparison according to Spearman's row, a pairwise comparison between gene expression profiles here in a huge matrix of hierarchical clustering of gene expression levels, then for the protein coding genes, and we had more than 20,000 of those in our data set, we find a pattern where the gene expression profiles really nicely cluster according to, first of all, tissue type, and if one would zoom in there, more or less according to phylogeny. This is much less so the case when one looks at non-coding RNAs. We have looked at long non-coding RNAs, the sort of largest class of non-coding RNAs in the cichlid's genome that probably play an important role in gene regulatory processes, because here these differences are much less pronounced, which is sort of also very significant statistically. But we use these data now to again use the same kind of reconstruction procedure that I have shown you before, namely the one where we sort of time slice the phylogeny and ask for evolutionary rate changes throughout the adaptive radiation of cichlids in Lake Tanganyika. And we do, in fact, see that the rate of gene expression evolution changes in the course of the adaptive radiation. For protein coding genes, it becomes accelerated towards the end, where also the number of lineages that emerge increase. But maybe much more interesting is really this observation that sort of the increased rate of gene expression evolution in the non-coding RNAs more or less matches the curve of the accumulation of distinct species that could point really to gene regulatory processes being the major genomic drivers of diversification, or drivers, I shouldn't say drivers, I should say the major correlate in an adaptive radiation. So to summarize my presentation of today, I have tried to show you that fishes, cichlid fishes are highly diverse across various levels of biological organization, from taxonomy to ecomorphology, but also in their genomes. What we found for the cichlid adaptive radiation in Lake Tanganyika is that the diversification was not gradual in the course of the radiation, but that it was characterized by discrete pulses of accelerated phenotypic evolution, and these pulses of accelerated evolution are compatible with an existing model of adaptive radiation, namely the so-called stages model, that has postulated that within an adaptive radiation, in a first stage, macro habitats should be exploited, which is compatible with our body shape data, then micro habitats, which would fit sort of diversification in the feeding related structure, and finally a phase of diversification along the signaling axis should occur, which could be sort of shown by pigmentation pattern differences, whereby in our case we clearly saw that in this final phase, so it exists also diversification in the pharyngeal jaw apparatus, so a feeding related structure occurs. And finally, I was showing to you that species-rich lineages are genetically more diverse as by their per genome heterozygosity, but also that the gene expression changes throughout evolution relate to an accumulation of species in the course of the radiation. And with this, I would like to come finally to the introduction of the team members that were part of this endeavor and without whom that could not have been worked out. First of all, Fabrizia Ronco, whom I mentioned in several contexts, she was leading two of the studies that I've presented here today. Atime Deltaya, who was working on the gene expression profiles, I've presented her results, and Michael Macina, who is in the meantime became professor in Oslo, worked on the genomics timeline. Also involved in all these three studies, main studies that I presented, was Astrid Böhne, who is now at PI at the Museum König in Bonn. I didn't have time to talk about her main results on sex chromosome evolution that I know some of the organizers might be very much interested in. Lucas was involved in the fieldwork of some studies, but also he had a big project in the context of our Cichlid X work, namely on the community structure. I didn't have time to talk about that. I would like to thank a lot Nicolas Boileau, our fabulous lab technician, Heinz Büscher, who went with us a lot of times to Africa. He did all the scans on the 2D morphometrics and shared with us all his secrets about Lake Tanganyika and the locations of type specimens and stuff. Then Adrian, our major fish expert in the lab, who spent together with Fabrizia and me all the time of field work in Africa. Anna Boiler, who did the preparations and analysis of the stable isotope data, and then Milan Malinsky and Virginie Ricci, who were involved in the bioinformatics part of the project. Not always possible within the lab, so we seek advice from two other people from the neighboring institute, Ansgar Kamen, who run the stable isotope samples, and Cichlid from the University in Oslo, who oversaw the genome sequencing project of the 500 plus Cichlid genomes. Of course, this project would not have been possible without generous funding from the ERC and some other funding agencies. And especially, I would like to thank many people in Africa, along Lake Tanganyika, in villages, in administrative offices and so on, without whom we could never have been in the position to collect all these different specimens. I would also like to thank some colleagues that have contributed with some samples. And with this, I would like to thank again Julia and the organizers for inviting me to that seminar, and for giving me the opportunity to present our Cichlid work to you. And with this mask from Africa, I would like to close my presentation, and I would like to thank everyone out there. I do not see any one of you, but I assume that maybe a handful of people have watched that, so I'll thank you very much for your attention. Thank you very much.
Thank you very much for your talk. That's very impressive work. While we wait for a few more questions from the audience, I was wondering how much is known about past environmental fluctuations or specific events in Lake Tanganyika that would correlate with the trait-specific pulses and accelerated phenotypic evolution that you observe?
Thanks, Julia. Yeah, that's an open flank to some extent. I mean, of course, we do know that Lake Tanganyika in its history of 10 million years underwent fluctuations. It never felt dry. It's a relatively stable lake system, also because of its positioning in the East African Rift Valley. It's very deep, but of course there must have been environmental fluctuations, but the resolution of the geological reconstruction of Lake Tanganyika is not as precise yet in order for us to compare it to these pulses that we see. However, there is an initiative has kicked off actually to start a scientific drilling campaign at Lake Tanganyika. So to drill the sediment all the way down and take all kinds of environment and geological and whatever proxies into consideration. So we hope that in the future we'll be able to link perhaps, or to test for a potential link between these pulses of ecomorphological diversification and external/environmental events. For the moment, it would be pure speculation, but we hope that, say, in about 10 years from now, we might know exactly that.
That would be very, very interesting. We have quite a few questions coming in now. One of them is, why are the radiation pulses not reflected in the non-coding RNA data? Would one expect RNA signals in the different tissues to match the timing of the pulses for the different morphological traits?
Perhaps that's a good hypothesis, namely that if we would know the genes that make the phenotypic differences, then they should actually match these pulses. But of course, what I've been showing is really the global picture of, say, all protein coding genes, all 27,000 genes, or the signal of all long non-coding genes. And at least if we look at that overall, we don't see this kind of pulses. But of course, we also don't know exactly yet maybe the genes that underlie these different traits. So that would perhaps, or would obviously be a next step to also sort of identify the genes that maybe make the pharyngeal jaw long or short or with more teeth or whatsoever, and then look or reconstruct the expression of those genes. But without the knowledge of the genes that underlie a trait, obviously the sheer amount of the genes that are out there, they overlay the signature of these genes. But it would be an expectation to find these pulses in those genes underlying certain traits.
What do you think is the reason for the correlation between polymorphism and diversification rate? Is it that high polymorphism indicates a large size of the ancestral population? Is polymorphism in species-rich tribes unusually high frequency, or is much of it shared between species?
A good question. It's, I guess, a combination of everything and difficult to disentangle. So heterozygosity is a straightforward measurement in the genomes, and we determined it as a genome-wide measurement. But what we do find in the Lake Tanganyika cichlids is that the range of per genome heterozygosity or per species heterozygosity covers the entire, pretty much the entire range of heterozygosity that is known across animals. Hannes Wadal and Milan Malinsky have a review paper out of that. So in our case, it's a correlation, and that the species which tribes have a higher rate or higher degree of heterozygosity at the per genome level. How it is intertwined with population sizes and so on, this we have not tested so far. For this, we also had too few genomes per species available, but as I tried to explain, what seems clear from simulations that we have been doing, it's a combination of gene flow between species plus sort of phylogenetic history of the different clades that all together explain more or less the pattern that we see. But this is clearly something that will need to be looked up into much more detail in the future. And the route of looking into population sizes is really something that that we are kind of up to.
Thank you for that. What do you consider are the determinants of genetic diversity in the tribes present in Lake Tanganyika? Is this higher genetic diversity associated to specific regions in the genome, or is it scattered throughout the genome? And I guess as the follow-up, like a second question, do you consider that processes such as plasticity could be associated in the explosive radiation seen in cichlids?
Maybe let's start with the plasticity question. Well, there is phenotypic plasticity in cichlids. There's phenotypic plasticity, for example, also in the pharyngeal jaw apparatus. Axel Meyer's lab, for example, and he himself during his PhD has worked on that. But in terms of this broad scale pattern and the morphospace that we have observed, I don't think phenotypic plasticity can explain much of that. And all those sort of plastic components that have so far been found are visible, but the range of the phenotype that is sort of plastic in a sense is really small as compared to the big morphospace. Or in other words, while in the pharyngeal jaw, depending on diet, for example, phenotypic plastic change can be invoked. This change is really small as compared to this between species differences that we find. So phenotypic plasticity may help in adaptation, local adaptation, or in mildly changing environments, but I don't think on the large scale it will do the job to kick off an adaptive radiation. Now, what are the determinants of diversity? Well, that's a big question. And this is something where we do not yet have a clear answer. Because there's so many different parameters that would need to be considered, but with cichlids in particular, we are starting to accumulate all these different kinds of information from genomic features, be it mutation rate or population size, all the way out to morphological or life history data. And I think the combination of those will ultimately allow us to address this question. So far, I think we cannot say anything about that, honestly.
Is there any information on how early in the life cycle are the key morphological differences seen, and when do they have functional relevance?
Well, this is, of course, another blind spot that the one that asked this question has seen in our work, namely that all the data I've presented today are adult data, adult only data, so the developmental component is missing from that. Well, I think it would be important to go along the developmental axis, let's put it that way. So in principle, the pharyngeal jaw apparatus, the oral jaw apparatus, also body shape to some extent, perhaps less so the pigmentation pattern, or in another context, they act at a relatively early stage of life, or must act in these cichlid fishes. Pharyngeal jaws are observable already at almost larval stages. But we don't have much of an idea about the relative function of these traits at early stages in life. This is also something that will need to be explored in the future.
Yeah, that would be very interesting to look at. And I think we have one more question about the role of introgression in adaptive radiations. It's true that there seems to be no correlation between F4 and number of species, but the amount of gene flow required for speciation or diversification may not be the same for each species pair. Also, do you think from your experience in cichlids that gene flow acts to deter diversification?
Thanks for that question. So with the genome data in hand, it's and not only of the Lake Tanganyika cichlids, but of other cichlids as well, or other cichlid adaptive radiations as well. It is evident that gene flow happens between species, especially within adaptive radiations, and at least for Lake Victoria and Malawi, a few smaller cichlid radiations, there is data out that suggests that hybridization at the onset, or even before the onset of the radiation, sort of laid the ground by reshuffling existing genetic diversity for radiations to kick off. For Lake Tanganyika, we don't have, based on our genome data, a signature of ancestral hybridization as a driver of diversification. But as I said, that the pattern of greater per genome heterozygosity, which correlates to species richness, is in part explained by gene flow between closely or more closely related species. That could, in a sense, hint towards a more or less autocatalytic process of diversification. Once you become a bit more species richness, you become more so because you have more partners to share genes with and so on. But I guess that remains speculation at this stage. So hybridization is a common feature in adaptive radiations. In some adaptive radiations, there's strong evidence that initial hybridization creates the substrate for radiations to take place in the first place. In Lake Tanganyika, we don't see that at the beginning with our data, but we do see gene flow throughout the radiation. How that might boost or have completely the opposite effect on diversification is an open question, but it's ongoing, and there's even within Lake Tanganyika, there's one species described that we sequenced, and it's an F1 hybrid, and that's all what we have seen so far. So it seems this taxonomic species is recreated over and over as a F1 hybrid, so also that might exist, in which case nothing happens further.
Thank you very much. I think we have one last question coming in, if that would be okay. To what extent, if at all, do you see species analogs between lakes, maybe similar across multiple traits that we might not intuitively expect among members of broad ecotypes?
Mm-hmm. A very good point. In fact, this is a line of research that I also didn't present, but that we are very much interested in our lab and in some other cichlid labs, because there's a lot of convergent evolution between cichlid adaptive radiations, but also within cichlid adaptive radiations. There's some famous examples of cichlid species in Lake Malawi and Lake Tanganyika that look very much alike. So they have evolved independently at a different time scale towards very similar phenotypes, not only in body shape, but also in color and pigmentation pattern. There might be many more cases of convergence in characters, for example, the jaws or whatsoever, that are not so obvious. It's a common feature with the idea perhaps that sort of the same environmental pressures lead towards convergence. Alternatively, there might simply be also the case that perhaps at some point cichlids have reached the amount of different phenotypes that they can produce. So you have too many species for what is possible to make phenotypes, which would also lead to convergent evolution. But the short answer to that question is there's a lot of convergence between adaptive radiations of cichlids, but even within adaptive radiations of cichlids, there is convergence when sort of two species that come from two independent lineages within the radiation converge in a literal sense into the same ecological niche within a lake. Again, perhaps because there's maybe not enough ecological niches in Lake Tanganyika.
Great, thank you for that. I think unfortunately this is all the time we have for today, but thank you very much for your talk again.
Thank you very much, and thanks for the great questions.
Next Wednesday, our speaker will be Dr. Siobhan O'Brien from University of Liverpool with her talk entitled Ecology and Evolution of Microbial Social Lives. Thank you all for watching, and we hope to see you again in the next one. Goodbye.